See, slowly people will come, doesn't matter. People work hard. See, when I started CSIR, there was only 2 students when I started in the first time, 3 years back. Now there are 400 students in the online. Okay, so people come slowly, don't worry about that. Okay, now, okay, let's quickly recall. In the previous class, we have completed what? You also please try to watch the previous classes, whichever you missed. Okay, so what we have completed, anyone remember? We completed C3 pathway, C4 pathway and C2 cycle. And we are left with the CAM pathway. Okay, so first in this class, new girl is there. Now I'll complete the cellular respiration first. This is very small. Okay, 10 minutes we can complete this. Okay, I'll quickly ask you simple questions. Answer: whenever RUBP combined with the oxygen in the presence of the Rubisco, how many stable intermediates we get? 2. How many stable? 1. Very good. Why I use the word stable? Stable means which is 3 carbons. So how many stable we get? We get only 1 molecule of 3PZ and 1 molecule of 2PZ. Very good. 1 molecule, it is not it. How many carbon it is? 2PZ is how many carbon? 2 carbon. And this will be? 3 carbon. This is very, very clear. What are the various type of these cell organelles that are involved in the C2 cycle? Chloroplast, peroxisome and mitochondria. Correct na? What is called Kranz anatomy? How many cells are involved in Kranz anatomy? And mesophil as well as bundle sheath cell. Now slowly think and answer. In case of a C3 cell, tell me Aisha, where C3 pathway is taking place? Which cells, mesophil or bundle sheath? See, in the C3 and CAM pathway there is only mesophil. Why? Because they do not possess Kranz anatomy. Hence C3 mesophil cell, CAM also mesophil cell. Correct na? And where the C2 cycle occurring? In a specialized organelle. Correct na? And last and final question, how many ATP are required or how many ATP are utilized in the C4 pathway? Slowly, okay? In the C4 pathway for 2 glucose molecules. Slowly. For C4 pathway, how many ATPs are consumed for 2 glucoses? Okay, tell me Sanjana, for C3 pathway, how many ATPs are required for 1 glucose? Forgot everyone. 18 ATPs. Ma, for 1 glucose we read 18 ATP. How many NADH? 2 NADH. And 6 CO2. Correct na? 6 CO2. This is very clear. Yes. And what we discussed in the C4 additionally, how many ATP? Ma, C4 we read na plus 12. Total? 30. At least recalled or not? You are not reading. 30 ATP we required. Why this 12 ATP extra? Anyone remember? Why? Because pyruvate, whenever it is converted into the phosphoenol pyruvate, the enzyme involved is dikinase. So pyruvate convert phosphoenol pyruvate 6 times. Then 6 into 2? 12 ATP molecules. So there we add that extra 12. Okay. Once again, remember my words very clearly. It is not important to read. It is important to remember. Focus on the remembering rather than reading. Okay. All right. Now, we'll complete the CAM pathway at the end. Small topic it is. Just 5-10 minutes it will take. So, after the photosynthesis, the second subtopic is cellular respiration. In the entire plant physiology, the toughest topic is this only. Most of the students cannot answer the questions from the photosynthesis and cellular respiration. Okay, 90% all the questions comes from statements or assertion reason or matching based questions. There are 3 possibilities majorly we will get it. Okay, and this concept I can tell to all of you, 10% to be remembered, 90% to be understood. By that you can answer any kind of a given question in the exam. Now tell me all of you, tell me Kusuma, what is the difference between respiration and cellular respiration? What is respiration? No, I am asking normal respiration. Exchange of gases. What we inhale? We inhale oxygen, we exhale carbon dioxide. Exchange of gases is said to be the respiration. What is cellular respiration? Very good. Cellular respiration is a process of oxidation of biomolecules. What are those biomolecules? Proteins, lipids, carbohydrates. Why we have to break them? To produce energy. What is that energy? ATP and NADPH. Okay, in very simple words. Okay, in this chapter we have to majorly focus on glycolysis, fermentation, Krebs cycle, electron transport chain and oxidative phosphorylation. Then energy related means how many number of ATP, NADPH are released, what is called amphibolic pathway and what is respiratory quotient. This is the exact syllabus. Okay. You strictly read PPT, you can answer all 100 questions. However difficult the question may ask, you can easily answer. Okay. Now, all of you simply give me the definition. See here, based on the location, respiration can be external, internal or cellular. Correct na? Now, in the respiratory system of human physiology, we will read what is external respiration and what is internal respiration. So, these two are talking about exchange of gases, right? These two are extremely different with cellular respiration. Cellular respiration is exclusively happening in the mitochondria, also in the cytosol. So, in this cellular respiration, whether we are using the oxygen or not, based on utilization of the oxygen, we categorize it into aerobic and anaerobic respiration. So, as the name itself indicating very clearly, aerobic respiration compulsory require the oxygen. So, compulsory require the oxygen. By using that oxygen, we are oxidizing something. What is called oxidation? Breaking down. Why we are breaking down something? To produce energy. And what is anaerobic respiration? Again we are breaking something to get the ATP, but we are breaking down completely in the absence of oxygen. That is called anaerobic respiration. Now, how internal external respiration differ with cellular respiration? Look into this. See, what are the various biomolecules we are having? Carbohydrates, proteins and fats. Okay. Now, if I ask you a question, among the carbohydrates, proteins, fats, which we use more oftenly for the energy production? See, which is their first source of energy? Carbohydrate. Correct na? Why carbohydrates is the major source of the energy? You take a brain. Brain only takes glucose as their major source of energy. If we did not eat anything, we obviously fall down. Why? Because brain is not receiving enough glucose to control and coordinate all the activities of the body. Correct na? So, any cell wants to be functional, it generally requires the presence of glucose. Correct na? And that glucose we majorly takes from carbohydrate. See, by breaking down the proteins also we can get the glucose. By breaking down the fats also we get the glucose. But brain or body generally uses carbohydrates as the major source of the energy or first source of energy, followed by proteins and fats. It's very simple process. Now, if I ask a simple question to all of you, we'll read in the upcoming slide. Which will give more energy, carbohydrate, protein or fat?
Carbohydrates.
Once again, let me ask you the question in this way. Among carbohydrate, protein and fat, which gives more ATP or more energy? Lipids or fats gives more energy compared to the carbohydrates and proteins. I think in a more simple way, okay. You had a bowl of rice. Rice generally contain what? Carbohydrate. Let's consider you ate a paneer pizza. Okay, that also contain protein as well as the more fat also. Okay, which will give more energy? See, always lipids gives more energy. You are getting the point. In terms of ATP calculations I am telling about. But first source of energy is carbohydrates. There is a simplest difference. Highest amount of the energy given by lipids. Primary source of the energy is carbohydrates. You are getting the point. Do not get confused over there. So, tell me finally how can I define the word cellular respiration? Cellular respiration is a breakdown of respiratory substrates. What are respiratory substrates? Carbohydrate, lipids, proteins. They are called as respiratory substrate. Oxidation or breakdown of the respiratory substrate to liberate or to get ATP. That is called cellular respiration. So, the cellular respiration or the aerobic respiration broadly divided into 4 steps. Glycolysis, Krebs cycle. Okay, between the glycolysis and Krebs cycle, something called as link reaction. Okay, we actually cannot consider it as the major step, but we will discuss that. Then electron transport chain, and there is one more process named as oxidative phosphorylation. By this process only we are going to produce abundant amount of the ATP. First one, link reaction, Krebs cycle, electron transport chain, oxidative phosphorylation. I will tell you in a very simple words, the ultimate goal of the cellular respiration is breakdown of which molecule? Glucose. Why only glucose? Whether fat breakdown or protein breakdown, amino acid breakdown, carbohydrate breakdown, ultimately what we take is glucose. Correct na? So in a simple words, cellular respiration is a process of breakdown of a glucose to get abundant amount of ATP. Example 36 to 38 molecules of ATP. So you cannot get 36 to 38 molecules of ATP directly by using one glucose. This glucose has to undergo all these steps. Then only we get the abundant amount of the ATP. Otherwise we never get it. Now look into this. Okay. Now I want you to understand this, but oh, where is the picture? Just a minute. Okay. Now all the students please look into this picture. Okay. Tell me, Ayesha, what is the definition of the metabolism? Sanjana, what is called metabolism?
Sum of chemical reaction.
Okay. Sum of chemical reaction which contains, see, I'll tell you one thing to all of you, listen. Whenever something is written in any book or in NCERT level, whenever you are reading some line, you have to apply or you have to understand, otherwise you never learn. What you are all trying to read is whatever written in the book, you are trying to read it. Instead of reading, try to apply it. Okay, then biology becomes so easy. Now what is called metabolism? You have said that metabolism is the series of catabolism and anabolism. That is called metabolism. Metabolism occur in every cell. Let's consider I have eaten a food. Just now I have eaten food. Food contain carbohydrates, protein, lipids. Now why I am eating the food? To get energy. Very good. To get the energy or to produce the glucose. By finally glucose is going to give rise to what? Energy only. So basically I am eating the food to get energy. Whenever I eat a food, I never eat the food in a simple form. I always eat the food in complex form. Correct na? Suppose I eat a rice or I eat a roti, which generally contains starch, which is complex form. So that complex form I have to break down into simple form. Then only I can get the energy. So that breaking down we call catabolism. In other words, breaking down is called oxidation. Oxidation of carbohydrates, fats and proteins. Called as catabolism. In simple words, catabolism means breaking down or oxidation of biomolecules. Why? To get energy. And why we need energy? Any cell never be functional without energy. How many cells are there in a typical human body? Okay. How many cells are there in bacteria? Bacteria, ma. Bacteria is single celled, right? Unicellular means how many cells? Only one cell. Whether it is a single celled organism or a typical human body like typical organism like human contains 37.2 trillion cells, not billion, trillion. Whether it is a single celled organism, whether it is a multicellular organism, the cell wants to perform any function. What does it require? ATP. Correct? Any function. So by using this ATP I am performing all the function. which are called anabolic functions. You are getting? So an example, I want to move my muscle, I require energy. If all the enzymes in the body to be functional, I require energy. If the cell wants to be divided, require energy. So catabolism is what? Breaking down. Anabolism is building up, to make something, to produce something, to synthesize something. That is called anabolic. So in the anabolism, what we are doing, we are spending energy. You are getting? In catabolism, we are producing energy. Anabolism, we are? So sum of catabolism and anabolism together called as metabolism. So in our body, carbohydrates break down, protein break down, and lipid break down. Finally, they give rise to ATP or energy. That is what we have to read in this entire chapter. Correct? So look into this. These are all the various type of biomolecules which we have to break. Look into the point. When I eat the food, it contains polysaccharide, lipid, and proteins. Everyone please look into this picture correctly. Now polysaccharide, example of polysaccharide, starch. What are the example of polysaccharides? Starch. What are the examples? We read now first year. Cellulose, glycogen. These are all said to be what? Polysaccharide. Poly means many. Saccharide, sugars. Many combination of monomers combined together and give rise to a polymer. Leave it. Okay, I'll tell you in the biomolecules. Now this polysaccharide such as starch. Let's consider starch. They break down into finally monosaccharides such as glucose, fructose, and galactose. But our body uses what? Glucose. Correct, na? Finally, all the monosaccharides ultimately converted into glucose. Finally, glucose undergoes what process? Glycolysis, and giving rise to pyruvate. Simple. So how we are getting the pyruvate? By breaking down. So which is called catabolism, also called as oxidation. Oxidation of glucose is going to give rise to pyruvate. Now pyruvate, we'll discuss that. I am just giving overview. Pyruvate in the presence of oxygen is giving rise to acetyl-CoA. Now acetyl-CoA enter into and ultimate function is produce ATP. Clear? This is the fate of carbohydrates. Now let's consider fate of protein. Proteins break down into amino acid. Amino acid contain a mechanism called as a what is called transamination? My name itself indicating that transamination. Transfer of amino group. See, this is how amino acid look like. Right side carboxyl group, or left side amino group, one hydrogen and one R group. This is what amine. So transfer of amino group is called. So upon transamination, amino acids are also going to give rise to pyruvate and acetyl-CoA. Again, amino acid is going to give rise to acetyl-CoA. Acetyl-CoA enter into citric acid cycle. Simple. Now we also eat lipids. All our vegetable oils are what? Lipids only. Na? Now lipids also break down into and glycerol. The question which is asked in the NEET exam, which of the following is not a polymeric in nature? What is the question? Which of the following is not polymeric in nature? What is called polymeric? Many monomers combine together and form the polymer. They have given DNA. I am giving examples. They have given cellulose, they have given glycogen, they have given lipids, they have given starch, and they have given protein. Tell me, which of the following is not polymer? Now what is polymer? Many monomers come close together and give rise to polymer. Tell me, you all people, protein made up of amino acid. So amino acid can act like what? Monomer, one unit. So many monomers give rise to protein, hence protein is polymer. DNA made up of nucleic acid itself, DNA. Nucleic acids are DNA and RNA. So DNA made up of nucleotides. Correct na? Many nucleotides come close together and give rise to DNA. So one nucleotide is monomer. Many nucleotides give rise to polymer, DNA. So DNA is also polymer. Cellulose is present where? Plant cell wall is made up of cellulose. Cellulose is generally made up of what? Glucose. Many glucoses come close together, give rise to cellulose. That is also polymer. Then glycogen, which is present in our body, that is also made up of Glucose. See, when I eat a food, excess glucose can be converted into the glycogen and stored in the liver. So glycogen is also made up of glucose. It is also polymer. Starch is also made up of glucose only. So among all the biomolecules, lipids are not polymers. Why? Lipids are made up of two different monomers. Got the point? One is glycerol and one is fatty acid. That's why it is not the polymeric in nature. But what is the ultimate goal? Whatever the biomolecule it may be, ultimately we have to get energy. So glucose given rise to pyruvate by what process? Glycolysis. Very good. So breakdown or oxidation of glucose is called as glycolysis. Similarly friends, breakdown of fatty acids called as beta oxidation. That is also oxidation. The process called as beta oxidation. Now friends, we also have a glycerol. Glycerol also going to give rise to pyruvate. Pyruvate is going to give rise to. So can you tell me one very common point here, or can I say like this? Whether it is a carbohydrate, lipid or protein, everything is going to give rise to. Finally acetyl-CoA entered into the. So we can say that acetyl-CoA is the major intermediate molecule which is coming from all the biomolecules. Finally, any biomolecule, in order to produce the energy, they have to enter into. If the citric acid cycle wants to start, we require. Without that, we cannot start this citric acid cycle. Don't get confused over there. So it is very simple. Now, in the exam, I already told you, in the photosynthesis, most of the questions comes from locations where the light reactions of photosynthesis occur. Membrane or lamella. Dark reactions, stroma. Why? Because enzymes located over there. Similarly, what are the 4 steps of cellular respiration? Glycolysis, citric acid, etc. You can call it oxidative phosphorylation or chemiosmosis. We read the same chemiosmosis in photosynthesis, which is required to synthesize. Same here also we are doing the same. We also called as oxidative phosphorylation. So directly they will raise the questions in the exam where all these processes are happening. Glycolysis strictly happens in the cytosol. Direct question. So glucose converted into in the cytosol. Very important statement. Now pyruvate is going to give rise to, just now we read na, acetyl-CoA. This is the most important. This is exactly called link reaction. We read extreme depth in the upcoming slides elaboratively. Okay. Okay, yes. Okay, now look into the point very correctly. So pyruvate converted into what?
Acetyl-CoA.
Now the question asked in the exam, where? Where pyruvate converted into acetyl-CoA? Mitochondria where? Mitochondrial matrix. What is mitochondrial matrix? We know, no? This is outer membrane thrown into foldings called as cristae, and this space is called matrix. Exactly in the matrix, pyruvate converted into, then acetyl-CoA entered into the Krebs cycle. Where the Krebs cycle is happening? Very good, mitochondrial matrix. After that, we have electron transport chain that happens in the inner membrane of mitochondria. Here, can you see that? Cristae. Why it is only happening over there? Because inner membrane of mitochondria having large surface area. We read now 4 electron transport enzymes are there. Complex 1, complex 2, complex 3, 4. Where they are present? They are present on the inner membrane. So electron transport chain exclusively happens on the inner membrane. Then the last and final process called as oxidative phosphorylation or chemiosmosis. It occurs exactly between intermembrane space to the inner membrane. So this is outer, this is inner, exactly here it will occur. Correct, na? So by using these 4 processes, we break down the glucose and we give rise to energy. How many molecules? 36 to 38 molecules of the ATP. But we can't give it in a simple step. We have to undergo all these steps. This is what exactly the concept. Every single question comes from these 4 steps. Now let's try to understand number 1 step. What is that? Glycolysis. Tell me what is the ultimate goal of the glycolysis? Breakdown of glucose into pyruvate. Simple. So breakdown, also called as oxidation. What is our goal, friends? Our goal is by breaking down the glucose into 36 to 38 ATP molecule. But we can't get this much ATP in the glycolysis. It is the beginning step. We get a minimal amount of ATP. So glucose converted into the pyruvate, which means breaking is happening. So glycolysis is a stepwise. Why did we use the word stepwise? Glucose cannot break down into pyruvate in one step. It takes 10 steps. And 10 steps we require 10 enzymes. That's why glycolysis is a stepwise enzymatic breakdown of glucose into 2 molecules of, what is that? Tell me, what is that 3 carbon? Pyruvate. And this is always remember, glycolysis always occurs in the anaerobic condition. Always. Means in the absence of oxygen also we can produce ATP, but we produce very less ATP. That is not sufficient for the survival process. So what we require? We have to produce abundant amount of ATP, lot of ATP. If you want to produce lot of ATP, what do we require? Oxygen. You are getting. So only glycolysis is anaerobic. Remaining Krebs cycle, etc. Remaining all are aerobic. That's why we produce more ATP. Okay, now all of you focus on the picture. It is so easy to understand here. Just a moment, where is the slide? I think slide got missed. It's okay. It's okay, no problem. Okay, this important step is missed. Okay, no problem, I'll explain you here. First, what has to happen, friends? Glucose has to break down into pyruvate. Correct, na? Each pyruvate is how many carbon?
3 carbons.
Glucose is
6 carbons.
So how many pyruvates I get?
2 pyruvates.
2 pyruvates I get. And when the glucose is breaking down into 2 pyruvates, there is a production of 2 ATP. And there is a production of 2 NADH. There is a release of 2 water molecules. We'll prove it. So remember 2, 2, 2, 2. 4 2s. 2 pyruvate, 2 ATP, 2 NADH and 2 H2O molecules. No need to get confused, easy to understand. So now friends, let's focus the first step and tell me where this process is happening, friends? Cytosol. Correct, na? Now let's consider it is a plant cell or a animal cell. Where is the cytosol is located? Inside. Correct, na? Which means glucose, let's consider it's a plant. So glucose has to cross which one? Cell wall. Glucose has to cross cell membrane, cell wall, cell membrane. Then only it can enter into the cytosol. Now the question is here, how glucose can cross the cell wall and cell membrane? Glucose is, because we all read na, plasma membrane is impermeable or selectively permeable. Then how it will cross? On the plasma membrane, all of you remember, we are having a something receptors called as what receptor? GLUT receptors. What is that? GLUT. Glucose transporters. Okay, because of the presence of the glucose transporters only, glucose enters into the cytosol or they can cross the plasma membrane. So friends, the interesting point here, whenever glucose enters inside, immediately, look into the point, glucose immediately convert into 6-phosphate. This is a very, very good question. Why? Why it is converted immediately into glucose 6-phosphate? Because phosphate is what charged? Phosphate is negatively charged. See friends, plasma membrane never ever transport any negatively charged, any positively charged ions. No ion can transport directly through the membrane. So when the glucose convert into glucose 6-phosphate, glucose never go back. You are getting the point. So we are trapping the glucose inside. For that reason, glucose converted into glucose 6-phosphate. This is one of the most important reaction. All right, now let's study this particular glycolysis process. What is your ultimate goal of the glycolysis? Production of 2 pyruvates by breaking down the glucose. But friends, we can't break down the glucose directly into pyruvate. It contains total 10 enzymatic steps, 1 to 5 steps and 6 to 10 steps. All of you remember, if you ask me a question, sir, is it necessary to remember the structures? 90% it's not required. 10% if the question paper is really tough, there is a possibility that they will raise the question from the structure. So it is idealized to remember the structure also. Okay, now 1 to 5 steps of glycolysis is called as investment phase. 6 to 10 steps of the glycolysis is called as payoff, okay, or profit phase. Simple example, I want to start a business. What I require first? Capital, correct na? First I will invest the capital. Once if my business is going well, I will get back my capital. Along with my capital, I will also get profit. Correct na? So what is the ultimate goal of the glycolysis? We have to produce pyruvate plus ATP also. Correct na? So if I want to get the ATP, first I have to spend ATP. First I will spend. Then whatever I spend, I will get it back along with the profit. Correct na? That's why we call it investment phase and a payoff phase. No point of confusion. All of you focus on the picture very clearly. So first step, glucose is how many carbon?
6 carbon.
Converted into?
Glucose 6-phosphate.
Is there any change in the carbon number?
No.
Glucose is 6 carbon, glucose 6-phosphate is 6 carbon. Then what is the difference? The difference, at the 6th carbon, there is an addition of phosphate. That's all. Both carbon numbers are absolutely similar. Correct na? Now all of you remember, glucose converted into glucose 6-phosphate, which means you all told that there is an addition of phosphate group at which carbon?
6 carbon.
Now my question is, who will add that phosphate? Because, very good, who will add it? Enzyme. So always remember as a shortcut, the enzyme which add the phosphate called as kinase. Always remember, okay. The enzyme which removes the phosphate called as phosphatase. You are getting the point. The enzyme add the phosphate is kinase, removes the phosphate is
Phosphatase.
now glucose is what kind of a sugar? Pentose or hexose?
Hexose.
Hexose means 6 carbon. That's the reason the name given as hexokinase. Because the enzyme adding the phosphate to the hexose sugar. Now anyway, enzymes we'll see in the upcoming slide. Glucose converted into glucose 6-phosphate, no change in the carbon number. Glucose 6-phosphate converted into?
Fructose 6-phosphate.
That fructose 6-phosphate is also?
6 carbon.
Then what happened? Both are isomerases, which means both are interconvertible to each other. Fructose can convert it to glucose, glucose can convert it to the fructose. Hence, when both are interconvertible to each other, the enzyme involved is isomerase. We'll see it later, which can easily interconvert it. Now fructose 6-phosphate is how many carbon?
6 carbon.
Now fructose 6-phosphate, which is a 6 carbon containing compound, all of you look into this, converted into fructose 1,6-bisphosphate. Tell me friends, what is the difference? Fructose 6-phosphate is how many carbon?
6 carbon.
Fructose 1,6-bisphosphate?
6 carbon.
Count the carbon, 1, 2, 3, 4, 5, 6, there is no change in the carbon number. So, in case of fructose 6-phosphate, at the 6th carbon there is addition of 1 phosphate. When it comes to the fructose 1,6-bisphosphate, where we added? That's the reason we called 1,6.
Bisphosphate.
So, don't think that we added 2 phosphates. How many we added? Because already we added 1 here. To the which carbon?
6 carbon.
Here we added extra phosphate to the? Here who donated? See, here you all people said that kinase is the enzyme which added the phosphate. Imagine I am a kinase, this is the glucose. I am a kinase, I will just go and add here. But who will donate me? Who will donate phosphate group for me? Someone has to see, I am an enzyme, I will donate, I will add the phosphate here. But who is donating this phosphate? ATP. So every student think that enzyme itself donate the phosphate group. That never do it. I am enzyme, I will just add it. So this phosphate someone has to donate me. Who will donate? ATP. That's what we said now, investing. We have to invest it. So in the first step of the glycolysis, 1 ATP we spent. Correct na? Upon donating the phosphate, ATP hydrolyzed to?
ATP.
Correct na? Then in the third step what happened? Fructose 6-phosphate converted to?
1,6-phosphate.
Which means on the first carbon we added?
1 phosphate.
1 phosphate. So again enzyme comes is kinase. So someone has to donate the phosphate. Again ATP. So first step we spent ATP and the third step also we are spending ATP. Correct or not? Now what's going to happen now? Fructose 1,6-bisphosphate is how many carbon?
6 carbon.
No change in the carbon number. And in the fructose 1,6-bisphosphate, how many phosphates are there?
2 phosphate.
Now fructose 1,6-bisphosphate is unstable and it is breaking down into 2 molecules. Each molecule is how many carbon? Count the carbon number. 1, 2, 3. 1, 2, 3. And what else you can observe? Every 3 carbon added with 1 phosphate. Correct na? So fructose 1,6-bisphosphate upon hydrolysis, it is giving rise to? Very good. 2, 3 carbon containing compounds with each phosphate. Correct na? One is called as DHAP, dihydroxy acetone phosphate. Next one is called G3P, glyceraldehyde 3-phosphate. Where we read G3P?
C3.
C3. Correct na? That is also 3 carbon, but it do not contain phosphate, but it contain phosphate. Now till 5 steps are said to be what steps? Investment phase. So look into the picture. I'll explain everything briefly. Just don't worry about that. Glucose first converted to
Glucose 6-phosphate.
catalyzed by versatile enzyme hexokinase. So the function of this enzyme is what? Only adding the phosphate. But phosphate can be donated by ATP molecule. Glucose 6-phosphate converted to its isomer. Both are same by isomerase. Now fructose 6-phosphate converted to 1,6-bisphosphate again by?
ATP. Another phosphate.
Very good. So how many total ATP we use in the first step 1 and step 3? 2 ATP. Fructose 1,6-bisphosphate, which is a unstable compound, this one. Since it is unstable, breaking down into? One is glyceraldehyde 3-phosphate and another one is dihydroxy acetone phosphate. Now friends, no point of getting confused over there. I want you to understand the point very correctly here without confusing. Like this I can write DHAP and G3P are like this. What do you mean by that? Whatever the function performed by G3P, same function performed by DHAP. An example, G3P lost 1 carbon, it also lose 1 carbon. It gain 1 ATP, it also gains 1 ATP. While writing or while considering, we'll consider for 1 molecule. While calculating, we'll calculate for 2 molecules. While understanding, we'll understand for 1 to avoid the confusion. But while calculating, we'll calculate for 2. Don't worry. Okay. Now I'll explain it slowly. What happened to this? Okay. Now all of you focus. This is glyceraldehyde 3-phosphate. How many carbon?
3 carbon.
With 1 phosphate. Now all of you look into this. So DHAP also do same function. Correct na? So glyceraldehyde 3-phosphate converted into 1,3 bisphosphoglycerate. 1,3 bisphosphoglycerate is how many carbon? 3 carbon. 1, 2, 3 with 1 phosphate. Correct or no, friends? Correct or not? So 1,3 bisphosphoglycerate. So can I say one statement? If glyceraldehyde 3-phosphate given rise to 1,3 bisphosphoglycerate, which is 1 molecule, ultimately DHAP also give rise to 1,3 bisphosphate, because both will perform same function. Can I say a direct statement? I will get 2 molecules of 1,3 bisphosphoglycerate.
Yes, sir.
In simple words, now very important, this is a reduction reaction. What is that?
Reduction.
Tell me, we read the C3 cycle so clearly in the second step. In the reduction, what happened? We spent ATP and NADH also. Correct or not? Yes. So anyway, here we are gaining NADH, not spending. It is a reduction, which means when one glyceraldehyde 3-phosphate reduced to 1,3 bisphosphoglycerate, then 1 NAD plus reduced to NADH2. Simple. So, but how many molecules we produced? Two. Can I say 2 NADH2 are produced in the 6th step of glycolysis?
Yes.
Correct, na? According to the many calculations, sometimes they will always change it, but this is standard. 1 NADH always give rise to 3 ATP. Some books, but it is also true, they'll consider instead of 3, they'll consider 2.5. That is also right. You have to calculate according to the given options in the exam. Whereas 1 FADH2,
2 ATP.
this is also called, you remember my words in the photosynthesis I told you, these two are said to be reducing equivalence. Wonderful. So FADH2 gives rise to 2 ATP, in some conditions 1.5. 1 GTP gives rise to 1 ATP. What is ATP full form?
Adenosine triphosphate.
GTP full form? Guanosine triphosphate. Ultimately cell directly uses,
ATP.
that's why GTP also converted into ATP. We'll understand where we got this GTP, what is the story of this. Now, what I am saying here, glyceraldehyde 3-phosphate, one molecule give rise to one molecule of 1,3 bisphosphoglycerate. There is no change in the carbon number. This is 3 carbon, this is also 3 carbon, but it is what reaction? Reduction. Since it is a reduction, NAD plus reduced to NADH2. So I produce 1 here, I will also produce 1 here. So I am directly writing 2 NADH and 2 1,3 bisphosphoglycerate. Crystal clear. So now friends, 1,3 bisphosphoglycerate, how many carbon it is?
3 carbon.
Converted into 3-phosphoglycerate. What friends?
3 phosphoglycerate.
Tell me what is the difference. If you observe the 1,3 bisphosphoglycerate, first carbon it contain phosphate group, third carbon it contain phosphate group. Correct na? So 1,3 bisphosphoglycerate converted into, tell me why did I use the word bisphospho? 2 phosphate. Now why I am using the word 3 phosphoglycerate? So which phosphate is removed from which carbon?
First phosphate.
First phosphate from first carbon. That's why we called only 3 phosphoglycerate. Now we don't use the word bisphosphate. Correct na? So now can I say directly, when 2 molecules of 1,3 bisphosphoglycerate are going to give rise to 2 molecules of 3 phosphoglycerate. Correct na? So tell me what happened now? Someone lost the phosphate. So someone will gain that phosphate. Correct or not? Because phosphate means energy. So who will take that ATP? Sorry, phosphate. ADP. ADP combined with PI give rise to ATP. So can I say 2 ATP I will get?
Yes, sir.
Why? Because same function will happen here. This is which step? 7th step. So once again look into this. First glyceraldehyde 3-phosphate giving rise to 2 molecules of 1,3 bisphosphoglycerate, where there is a production of 2 NADH. Then 2 molecules of 1,3 bisphosphoglycerate lose the phosphate groups, convert into 2 molecules of 3 phospho. No, you have to use this 3 phosphoglycerate because we removed phosphate from only first carbon. Correct na? Then how many ATP we got? Total. Wonderful. So in the 7th step we got 2 ATP, we gain. And in the 6th step, 2 molecules of NADH. Correct na? Now 3 phosphoglycerate, how many carbon?
3 carbon.
Very, very good. Converted into,
2 phosphoglycerate.
so 90% of the students will think that there is a loss of phosphate group.
Phosphate group contains
Shifted. Correct na? So from the 3rd carbon, phosphate group shifted to 2nd carbon. So 3 phosphoglycerate converted into 2 phosphoglycerate. Correct na? Is there any change in the carbon number? No. Is there any gain or loss of energy? No. Very good. Now 2 phosphoglycerate is how many carbon?
3 carbon.
Wonderful. Converted into phosphoenol pyruvate. Tell me where we read this Aisha, phosphoenol pyruvate? C4. Very, very good. Correct na? Phosphoenol pyruvate is the first carbon acceptor. Correct na? So phosphoenol pyruvate is how many carbon?
4 carbon.
Phosphoenol pyruvate 3 carbon. Correct na? Oxaloacetic acid is 4 carbon. This is a 3 carbon. So can I say I get 2 molecules of phosphoenol pyruvate?
Yes.
Now, 2 molecules of phosphoenol pyruvate converted into 2 molecules of? Tell me what happened. What is the difference between phosphoenol pyruvate and pyruvate? Only difference is phosphate group. So it will also lose the phosphate group. So someone has to accept that phosphate. Who will not? ADP will accept. We get ATP. Can I say I'll get 2 ATP?
Yes.
Correct, na? So can I conclude? On the 6th step of the glycolysis, I get 2 NADH. 7th step I get 10th step I get 2 ATP. So if I tell you the simplest calculation, how we people can calculate this in a very simple words. In the first and third step of glycolysis, I have spent how many ATP? Correct, na? I spent. Whereas 7th step and 10th step, how many I gain?
4
Very, very good. I gain 4 ATP. Since I spent 2 ATP as my investment, I'll remove that. Then what I can get?
2 ATPs.
Okay, this is called net. Okay, net means, friends, in a simple words, remember, visible. Whatever you can see is net. But recall everyone, in the 6th step of glycolysis, how many NADH we produced? 2 NADH. We already discussed that according to our calculation, 1 NADH equal to 3 ATP. So we get 6. 6 plus 2? 8. 8 means, when they use the word 8, it is gross. What is that? Gross means total. Correct, na? But can you see 6 ATP directly in the form of NADH? No. Suppose example, simple words. I am having a 8 rupees check. Which indicates, do you think cash is there in my hand? No. I have to go to bank, I have to encash it. But if I show you 2 rupees coin, it is net, visible. So net is
2.
gross is
6.
8. Ma, gross means total. Gross means
Total.
you have total 8 rupees. In the 8 rupees you can see
2 as net.
2 rupees directly you can see. You cannot see remaining 6. So it doesn't mean that gross is 6. Total how much? 8. Out of 8 you can see 2. That becomes net. Most of the people will get confused and always answer gross means 6. Gross is
8.
net is only 2. Okay, don't confuse over there. I have written every single step very elaboratively. I strongly believe you all people answer the question. Okay, 2023. Slowly. You? What is the 4? But there is nothing to confuse, right? It is very clear. ATP is used at 2 steps. What are those steps? Step 1? Very good. Step 1. What is step 1? Glucose converted to glucose 6-phosphate. Third step? Fructose 1,6-bisphosphate. Wonderful. So assertion A is undoubtedly true. Reason says that first ATP is used in converting into glucose 6-phosphate, and second ATP is used in, it was only given, right? Fructose 6-phosphate to fructose 1,6-biphosphate or diphosphate. So these two are, both are correct. Very good. Both are what? See, that word is very important. When you get an idea that both are interlinked, that is the hint to understand that reason is very correct for assertion. When they are not interlinked, there itself you have to disconnect both. Both are completely irrelevant. Okay. So both A and R are true, and R is the correct explanation for the A. And what is this question? 2022. 25 you got more simple.
2, sir.
What is the question? Net. Correct, na? How much it is? 2. Okay. What is this?
Exokinase.
Slow, so this much faster, slowly. Hexo. Can you tell me what is this they have given here? What do you mean by that?
That membrane is not.
No. That is different. That is trapping. Glucose converted to the first reaction. Anyway, your answer is what? Hexokinase. Correct, na? Now let's read a most difficult concept. Okay, where every student will read 10 steps. They'll by heart 10 steps so nicely, and they'll leave remaining. If the question comes very tough, question comes from here. So how many enzymes are there total? Total how many enzymes in a glycolysis? 10. Correct, na? Out of 10 enzymes, there are 3 reactions which are irreversible. What is that? Irreversible. What do you mean by the word irreversible? You cannot bring it back. You're getting. And remaining how many are there? Remaining how many? 7. Which are called as? Reversible. See, this is not written in the NCERT. You have to know it so clearly. Now, what are those irreversible? Okay, why we called it as irreversible? What's the first step?
Glucose to glucose 6-phosphate.
Very good. Correct, na? Catalyzed by enzyme?
Exokinase.
Very good. Okay. Third step.
Fructose to fructose 1,6-bisphosphate.
Fructose 6-phosphate to?
Fructose 1,6-bisphosphate.
Correct, na?
Isomerase.
No. This is not isomerase. Glucose 6-phosphate converted. What is that? Wonderful. It is called PFK. Phosphofructokinase. Then what is the 10th step? Phosphoenol pyruvate converted to?
Pyruvate.
The enzyme is pyruvate kinase. Correct? Now what is that? Pyruvate kinase. So among the 10 steps, 3 are what steps?
Irreversible.
Why did I use the word irreversible? First, an example. Glucose can be converted to glucose 6-phosphate. This is only catalyzed by what enzyme?
Exokinase.
Enzyme. Without this enzyme, we can't convert glucose to glucose 6-phosphate. In very simple words, we can say that they all perform Gibbs free energy reactions. Okay, or in other words, if this enzyme is absent, no other enzyme can replace this. You are getting my point. If this enzyme is absent, you tell me, in the C3 plant, Rubisco is absent. Someone else can do the function. No one can do it. Entire reaction will stop. Same way, if this enzyme is absent, no one can catalyze this reaction. You are getting? That's why it is called what enzyme? Irreversible enzyme or irreversible reaction. Next, fructose 6-phosphate can be converted into?
Fructose 1,6-bisphosphate.
That is catalyzed by PFK. Phosphofructokinase. If this is absent, no one can perform. Phosphoenol pyruvate converted into pyruvate by an enzyme called as? If this is absent, again no one perform. Now the more difficult question compared to this, they will raise a question, which of the following are irreversible reactions, like however you see in the statement, the 1, 3, 10. If they can also ask a wonderful statement, which of the following is the rate limiting enzyme of glycolysis? Okay, if the question paper is so tough, they can ask this. Rate limiting enzyme. Now you all has to understand me, sir, why we use the word first of all rate limiting. Tell me friends, phosphoenol pyruvate converted into what?
Pyruvate.
Correct, na? Now I don't know whether you remember or not. In the first slide we read amino acid breakdown, protein breakdown and give rise to? Amino acid undergoes which process? Transamination. Given rise to?
Pyruvate.
Correct, na? Given rise to what? So can I say a statement, even though this enzyme is absent, but still I can produce pyruvate. Correct, na? You're getting the point. So even though it is an irreversible reaction, but it is not rate limiting enzyme, which means this product can be given by someone else. You're getting my point? Now see this, glucose can convert into?
Glucose 6-phosphate.
Now you tell me, in my body, consider I had lot of biryani, then I get lot of glucose, but my body never use the entire glucose. How much glucose is required? It use only that much. Remaining glucose is stored in the liver, muscle, little bit in the brain. Now tomorrow morning I am doing fasting. Then even though I am doing fasting, I can easily survive till the evening without eating anything. What's happening? The stored glycogen break down into
Glucose.
wonderful. First stored glycogen break down into glucose,
6-phosphate.
then
Glucose.
you are getting the point. So you tell me how glucose 6-phosphate convert into glucose? By removing,
Phosphate.
who will remove? Phosphatase. You remember, kinase adds it,
Phosphatase.
remove it. But what is my point here? By using, by breakdown of glycogen also I can produce glucose 6-phosphate. By that I can get the glucose. So my question, even though this enzyme is absent, I can produce glucose 6-phosphate. So maybe the step 1 and step 10 both are irreversible reactions, but the enzymes are not. So then fructose 1,6-bisphosphate cannot be produced by anything except PFK, phosphofructokinase. Hence, among the 10 steps, PFK1 is said to be the rate limiting enzyme. You got the point correctly. It is a very important statement you all people has to remember for the exam. Mandatorily, if the question is very tough, they may raise the question. So friends, now look into this. What is the first step? Which means glucose converted to glucose 6-phosphate. So the enzyme required is here what? Hexokinase. Again the question asked in the exam, which of the following ion can act as a cofactor for hexokinase, which is magnesium. You read the molecular basis of inheritance, na? DNA polymerase enzyme, that also required. You remember chlorophyll. Chlorophyll also required magnesium. So magnesium is one of the major cofactor, even in the plant also, even in the animal also. Very important it is. So what happened here? This phosphate group is added to the 6th carbon of the glucose, so converted to glucose 6-phosphate. Then what is the next step? What's happening here? Is there any change in the carbon number?
No.
Glucose 6-phosphate just converted to fructose 6-phosphate. Correct now friends? Tell me, what is this particular difference?
This form is reducing into another.
Okay, in very simple words, I will tell you in the biomolecules chapter. Glucose is a type of aldose sugar. You are getting? Fructose is a ketose sugar. Friends, glucose having how many carbon?
6 carbons.
The 6th carbon containing this group, aldehyde group. 5th carbon in case of fructose contains ketone. That's all the difference between the glucose and fructose. Okay, so what happened now? Aldose group converted into the ketose. There is no change in the carbon number. Since one can be converted to other, the enzyme is isomerase enzyme. We called as phosphoglucoisomerase enzyme. No point of getting confused here. What is the third step? Most important step. So how many phosphorylations are there? 2. That is important for the exam. 2 phosphorylations, step 1 and step 3. Fructose 6-phosphate added with ATP. ATP donate the phosphate group to the 1st carbon of the fructose, can be converted into 6-bisphosphate by a versatile enzyme, fructokinase, which is the irreversible and rate limiting enzyme. And magnesium act like
Cofactor.
very important it is. So they will ask a simple question, in how many steps of the glycolysis magnesium is required? No one can answer. See, so many are there, na? Okay. Now what is the 4th one? So finally what do we got? Fructose 1,6-bisphosphate we got. Correct, na? How many carbon? 6 carbon. It is unstable. We are breaking down, which is called or hydrolysis. Correct, na? Into G3P and DHAP. which is the very simple here. Okay. Then first 5 steps of the glycolysis are over. Then what happened to this? These two are isomeric in nature. Correct na? Which one? DHAP and G3P. Glyceraldehyde 3-phosphate, or you can also call phosphoglyceraldehyde 3-phosphate. Anything is same. Correct na? So 5th step only isomerization. So in 5 steps, how many phosphorylations are there? How many isomerizations? 2. Correct na? Glucose 6-phosphate converted to fructose 6-phosphate at the same time?
Fructose runs up 1 comma.
No. Fructose, sorry. DHAP converted to G3P. This is one isomerization, and another one is glucose 6-phosphate to fructose 6-phosphate. 2 isomerizations and 2 phosphorylation. Correct na? One is cleavage. Simple, easy to remember. Okay. Now the 6th step. What is the 6th step, friends? Formation of 1,3 bisphosphoglycerate. I want all the students to remember here. This is the most important. In all the 10 steps of the glycolysis, there is only one reduction step. Okay, only one reduction. So what we got, friends? NADH. Correct na? I'll give you a small hint to you. 95% of the cases, only 5% cases it will be varied. Wherever you see the production of NADH or FADH2, wherever you see, always remember the enzyme involved is dehydrogenase. Wherever you see production or reduction, wherever there is a production of NADH and FADH2, enzyme is dehydrogenase. Correct na? Here the enzyme is triose phosphate dehydrogenase. Tell me, why did I use the word triose phosphate? 1,3 bisphosphoglycerate is how many carbon? 3 carbon. That's why we can call triose phosphate dehydrogenase is the enzyme which is involved in the reduction reactions, which required to produce NADH or FADH2. And again the question, which require zinc as the major cofactor. What is cofactor, friends? What is cofactor? Cofactor called non-protein part. We'll read in the biomolecules. See, this is an enzyme, correct na? What is the general saying? All the enzymes are proteins, correct na? But the real point is here, 90% of the enzymes are proteins. What about the remaining 10%? We call non-protein part. You're getting my point? So we'll read it in biomolecules. The protein part is called as apoenzyme. You're getting? Non-protein part only called as cofactor. You got it? Again, cofactors are of different types. Coenzymes and prosthetic groups. That we will read it separately. Okay? You should have a very good knowledge. Okay? The next, what is happening in the 7th step, friends? 1,3 bisphosphoglycerate converted into? What happened that time? The phosphate group present at the first carbon got removed out. Correct or not? So then what is going to happen? Someone is, see, 1,3 bisphosphoglycerate is 2 phosphates. Correct or not? This phosphate is removed out. So someone has to accept, right? ADP is going to get accepted. There is a production of what? ATP. Correct na? So friends, this is a very important point to be remembered for the exam. This question also asked multiple times in the exam. When ATP is generated directly by phosphorylation of ADP during a biochemical reaction without involvement of system. What is called electron transport system? Electron transport chain means without using any complexes. Complex 1, complex 2, complex 3, complex 4 are present on the mitochondrial membrane, right? So we don't use any enzyme. You're getting my point? We don't use any enzymatic complex. If we get ATP directly from the ADP, that is called substrate level phosphorylation. Correct na? So at the 7th step we got substrate level phosphorylation. How many ATP we produced here? 2. Anyway, generally 1, but we'll consider for the DHAP also. In the 7th step we produced 2 ATP molecule. Very, very good. Now tell me, formation of 2 phosphoglycerate. What happened here?
The phosphate group
Is shifted from 3rd carbon to the 2nd carbon. No change over there. whenever there is a shifting occur, remember the enzyme involved is mutase. Correct na? Since the shifting occurred from phosphoglycerate, name of the enzyme is phosphoglycerate mutase. So simple. Then what is the ninth step? Tell me. Formation of 2-phosphoenol pyruvate. So friends, what happened here? 2-phosphoglycerate, very important friends, undergoes a mechanism of what is called dehydration. We should not say removal, okay? Loss. Decondensation means removal. I'm so sorry. Condensation means removal of the water. You are getting the point here. Dehydration means what? Loss. There is a difference between removal and loss. Correct na? Because of the dehydration, phosphoenol pyruvate converted in, sorry. What is that? What is that? Sorry, 2-phosphoglycerate, right? Yeah. 2-phosphoglycerate converted into 2-phosphoenol pyruvate. Just the reaction is dehydration. 2-phosphoglycerate is how many carbon?
3 carbons.
2-phosphoenol pyruvate? 3 carbon. There is no change in the carbon number. Now the most important here, again, the enzyme uses what? Magnesium. Okay, see how many times we use the magnesium. Okay, then finally formation of pyruvic acid. What happened? Phosphoenol pyruvate converted to pyruvate. What happened? Phosphate group got removed out. Phosphoenol pyruvate is how many carbon?
3 carbons.
Pyruvate is?
3 carbons.
There is no change in the carbon number. Just we removed the phosphate group. Correct na? So then what we produced? ATP molecules. So how many ATP?
2 ATP.
Molecules are formed. So the reaction is catalyzed by what enzyme? Again required.
Magnesium.
This is very important of the 10 steps. Correct na? So now you only see the final reaction, friends. Total ATP gained by substrate level phosphorylations are 4. Can you justify me? What are those 2 steps? No. Look into that. 7th and how? 10th. Wonderful. 7th and 10th. Ma, what I have written? ATP? Where we gained? 7th step and 10th step. So 2 substrate level of phosphorylations occur. So don't confuse. Total number of substrate level phosphorylations are 2. In 7th step we got 2 ATP, 10th step we got 2. So this no one will read it. You have to understand this correctly. Total number of ATP gained by the NADH are 6. Wonderful. Correct na? Now how many ATP we utilized in the step 1 and step 3? 2. Very good. Now let's calculate the net gain. Glucose. How many ATP? 2 ATP. Correct na? And 2 NAD. Finally what do we got? 2 correct no friends? Then we got 2 ADP, 4 ATP, 2 NADH and 2 water. Correct na? Anyway out of 4 whatever we spent we will remove it. Then we can say only 2 NADH, 2 water and 2 pyruvate. So 2 and 2. 2 pyruvates, 2 ATP, 2 NADH and 2 water molecule. Correct na? This is the first step of the cellular respiration which is called as glycolysis. Glycolysis is reduction or oxidation? What is called oxidation? Breaking down. How it become reduction? Correct na? What we are breaking? We are breaking what? Glucose. Now how it will become reduction? It becomes oxidation. Glycolysis is a partial oxidation of glucose, where 6 carbon glucose converted into 2 molecules of 3 carbon pyruvic acid. Friends, I am using the word net gain of 8 ATP molecules, but originally what is the net gain? Originally 2. Gross ATP is 8. Very good. Now we read in the starting there is aerobic respiration and there is anaerobic. Whether it is a aerobic or anaerobic, the common step is glycolysis. So in the aerobic glycolysis takes place, anaerobic glycolysis takes place. Aerobic and anaerobic differentiate from each other after the step of Glycolysis. You are getting the point. So in both steps we get glycolysis. Now you tell me, by using these two steps, where we get more energy? Aerobic only, correct na? Because anaerobic will stop there itself, because no oxygen. But here oxygen is there. After glycolysis also there are many steps will occur. But here it will stop with glycolysis only. Correct na? That's why anaerobic will get less ATP compared to the aerobic. See, this chapter, once I complete everything, you get a clear cut idea. Glycolysis is the only source of energy in the production of RBC, renal medulla. What is this renal medulla? Kidney contain? This is kidney? Forgot everyone, you all read na? What is that? Where the cortex and medulla are present?
In kidney.
Not in the kidney, in the nephron. Okay, okay, you can consider the kidney. Okay, so this is what a kidney structure. It contain what? Cortex, medulla. Correct na? So, but if the cortex and medulla wants to be functionally active, what do we require? We require the nephron. Kidney is never be functional. Functional unit of the kidney is what? Nephron. Okay. Any function performed by the kidney is mediated by the nephron. Okay. So, in the medulla, we have a nephrons, we call medullary nephrons. Cortex, we have nephrons, we can call the cortical nephrons. So, medullary nephrons, cortical nephrons uses ATP. To produce the ATP, they uses glycolysis. Brain and sperm. Why sperm require energy? Because sperm wants to move in the vaginal tract. Movement always dependent on what? Energy. So, all these ultimately starts using glycolysis for their major source of energy. Correct na? There is a simple question I will ask you. Is there any loss of carbon dioxide, or is there any release of carbon dioxide in the glycolysis?
No, sir.
In any reaction there will be no release or loss of carbon dioxide. Correct na? So what I am trying to explain you, all the carbons are preserved. You are getting the point. Glucose converted into 6 carbon, 3 carbon only. We have not lost even single carbon also. That's why there is a no loss of carbon or release of carbon dioxide in the glycolysis. All the carbon atoms are conserved. Okay, fine. This is the first step, that is the glycolysis. Okay, so question won't go beyond this level, but you have to read it. You people are not reading. Okay, without reading you can't remember. Okay, read multiple times. Okay, it's already 4:50. Okay, in the next class, first I'll complete the small camp pathway, then we'll complete the respiration. Let's consider by next Thursday all will be completed, entire plant physiology. What we do is complete Friday, you no need to take any test. I will show the questions here of PYQs first, then we will go to the practice questions. Then I will only revise the whole lesson in one class. By that you can remember it for a long period of time. Then we'll move to human physiology, the biggest unit. Okay, clear? Now remember, even though you play or you do whatever the activities, remember my words. In the NEET exam, self-study is more important. Okay, so we change the timing. So we'll start from morning 9 o'clock. Mostly by 4 or 4:15 we'll complete everything. Okay, sit up to 6:30. You have to go till which place? It is nearby only, na? It's taking time. 50. Why that much dear? Where are you from? Okay, it's taking 15 minutes of time. Okay. Anyway, make sure you'll reach home by 7:30 at least, because I want you people to read here. Okay, till 7 you be here. Okay, or 6:50. Okay, because you have to read at least 4 to 5 hours apart from the classes. I am talking about least. Okay, so classes, let's consider 6 hours, then preparation will be 10 to 11 hours, which is a kind of productive. Okay, and remember my statement so clearly, before going to the NEET exam, you have to read the whole syllabus more than 3 times. Then there is a possibility that you can get 450 to 550. Okay, if you revise more than 3 times. Okay, keep that in your mind and frequently revise it. Okay, done. Okay. Okay, I'll upload the notes. Immediately once you go home, you download this.